Okay, I would like to thank the organizers for giving me the opportunity to talk over here and to present our work in listeriosis. I'm going to present over here a cellular vaccine in listeriosis and a new antigen. The logical antigen in listeriosis, that is a bacteria that has been really broadly described and really well known all over, has been the main cytotoxin of the bacteria, that is listeriolysin. And I'm going to talk over here about a different antigen, that is the GAPDH. The GAPDH is the glyceraldehyde 3-phosphate dehydrogenase from Listeria, and I'm going to show you the data. Okay, this is Listeria. Listeria monocytogenes is a bacteria, a gram-positive bacteria, road, that is intracellular facultative, and is quite ubiquitous in nature, so you can find it all over in the silage, in vegetables, in the water, in animal assets, and in food. And in fact, food is the problem. You get contaminated food, and then you get infection with Listeria because of the contaminated food. Which kind of food? Well, the dairy products are the mainly contaminated food. Any frozen hamburger, any frozen stuff, vegetables, but also milk products, any cheese, any milk. But it's not a terrible bug. I mean, it's just important for a risk group of people, that is the immunocompromised people. Healthy people won't have a problem with Listeria. Even when you eat a lot of them, you only will have a really small diarrhea that it will be not different than any small salmonellosis or E. coli diarrhea, so you won't even notice that it's Listeria. But if you are an immunocompromised person, and by immunocompromised could be pathological immunocompromised people or non-pathological immunocompromised people, for instance, a pregnant woman is an immunocompromised person, that it will be quite affected by Listeria. So how is the cycle of Listeria? Oops, sorry. Listeria will expand in your bladder. It will go to the lymph nodes, and then from there to the bloodstream. And there, if your liver and your spleen didn't really get rid of the bacteria, then you will have a problem, because Listeria has a high tropism for the brain and for the fetus. And it will cause abortion in women and meningitis in small children. So this is the cycle, the inside the cell. In fact, Listeria monocytogenes is called monocytogenes because the main infected cell is monocytes. And this is the cycle in monocytes or in any phagocyte, dendritic cells, macrophages. There's a bunch of virulence factors well known from Listeria, the two internal lines involved in the entry of the bacteria, the GAPDH that is involved in the phagosome maturation, and the main important virulence factor that I was telling you before, that is listeriolysin. That is the toxin that it will break the phagosome and liberate the bacteria into the cytosol. And then in the cytosol, it has this funny thing that it was described for the first time in Listeria, but it's also common to other bacteria, like Shigella, that it makes comet tails of the actin filaments, and it gets a propulsion of the bacteria to infect the other cell without touching the extracellular space. So it's like putting a finger inside the other cell without touching anything outside, so without coming up outside the cell. And in this way, you will escape the immune system. And in the other cell, you will start back again the cycle of Listeria. Okay, so how does the immune system react against Listeria? Well, a typical listeriosis will be important first in innate immunity. The innate immunity is the non-specific immune response that is rapid. It withholds the infection. but gives you no memory. And in Listeria, the main important cells for innate immunity will be the phagocytes, macrophages, dendritic cells, neutrophils, and also NK cells. Then will come the adaptive immunity that is slower, but this will clear totally the infection, and it will give you memory. Cells involved mainly are B cells that produce the antibodies and T cells, CD4 and CD8. But in the case of Listeria, B cells are more important, meaning that the Listeria behaves like a typical intracellular bacteria, even when it is not. And antibody production is low and gives no protection at all against listeriosis. So you have to focus, if you want to have a vaccine against Listeria, you will have to focus in stimulating T cells, both CD4 and CD8. Okay, so if you want to make a vaccine against Listeria, then you will have to search for an antigen first, an immunodominant epitopes of this antigen, a vector, a vaccine vector, better non-toxic, better without the live pathogen, and then you will need to find the features of this vaccine to be efficient. Antigen-specific response, but also, and quite important in vaccines, also to trigger an important innate immune response. So, first point, check or search for a Listeria antigen and the immune-dominant epitopes. Well, Listeria, at the very beginning, it was not a pathogen, it was a saprophyte, and then something came up, that it was an induction of a regulator of all the virulence factors that came up, and then Listeria changed the life from a saprophyte to an intracellular life. And being in the intracellular life, if you want to make a vaccine that is effective against intracellular bacteria, you will have to focus in antigen processing route, because you will have both antigen presentation class 1 and class 2. So if you think on this, phagosomes will be ideal, because in phagosomes you will have antigen presentation by class 1 and by class 2 because of the cross presentation. So we were looking for antigens of Listeria that will be in the phagosome, and we came up with two. The classical, the listeriolysin, and the GAPDH. This is a classical, and this is very well known in LLO, which are the epitopes. Epitopes for the CD8 and binding to the class 1 is LLO peptide 91-99, and the classical one, CD4 restricted, is the 189-201. And if you look at the structure, the 3D structure of LLO, the class 1 epitope, it looks like a loop, while the class 2 epitope has an alpha helix. So when you came up with a new antigen, then you will have to think about this. How do we came up with the GAPDH as a virulence factor for Listeria? Well, we were studying the strategy of Listeria inside the phagosome, looking for the intracellular pathway and checking what was the strategy to stop the phagosome maturation. That means the stop fusing with the lysosomes. And we came up that the target of Listeria inside the cell was this protein, RAF5A. RAF5A is a small GTPase that is regulating endocytosis and phagocytosis. And when we started with a column with RAF5A, we came up with this protein of Listeria that we sequenced, and it turns out to be this LMO2459 that is the GAPDH. And when we check it, we map it where was the site of action in the whole GAPDH for inhibiting RAF5A, and we found out that it was in this small peptide at the end terminus of the GAPDH. And this small peptide is blocking the RAP5 through a new enzyme activity in this protein that is ADP ribosylating. When you ADP ribosylate and small GTPase, you block the ability to change from a GDP that is an inactive form to a GTP that is an active form. And that was this GAPDH from Listeria was doing. So checking for this small peptide, we thought that, well, let's see whether we can get antibodies just against this small peptide. And we did. So if you can make peptide antibodies without any adjuvant in a rabbit, then for sure you will have both types of epitopes, class 1 restricted and class 2 restricted. And that's what we started our study. Well, let's go and see whether we have a good vector to put this peptide. And we thought in two different vectors for a cellular vaccine, macrophages or dendritic cells. And we started, I have to say that both of them are bone marrow-derived macrophages or dendritic cells. So the first thing that we did is, well, let's see whether you can get infected with Listeria in the same way as all type of macrophages. And of course, using a mutant of Listeria that we know that doesn't grow inside the cells, that is the listeriolysin deficient mutant, you see that both macrophages and dendritic cells behave the same. Listeria grows inside, while the mutant doesn't grow. And next, we also check whether we can get processing of LLO inside those cells. And you can get processing by the binding of the processed form of LLO to the class 2 molecule, in both in macrophages and in dendritic cells. And also, we check for toxicity. And here comes our problem. It turns out that the macrophages, when they got either infected with Listeria or we were just using recombinant listeriolysin O and use it in the mice died. So it was really toxic to use this as a vaccine, while these ones were okay. So we start with this dendritic cell as a vector. So next, we selected the epitopes for our assays for the vaccination. And we started with something that we already know, with listeriolysin O epitopes, and we compare it with the GAPDH peptide, the 122. And you could check over here the bindings. You know, these were the binding to the class 1 of LLO189-99. Well, the binding of one peptide inside the GAPDH, it was in the same range. And when we check the class 2 binding, it was even better. You know, 75, almost 75 compared with 74. And we have even more than one just epitope inside this big thing. And when we check for the loop or for the helix of the class 2, we have it. So we thought that it was a really good peptide to try. So we did the vaccination with the dendritic cells and this GAPDH1-22 peptide. And we compare it, of course, with the LLO91-99 that it was already published, that it was working fine for protection against listeriosis. And if you could see over here, there's a decrease, so there's a better protection with our GAPDH. This is the classical listeriosis, that you have a huge splenomegalia and these granulomatous spots in the livers. This is the vaccinated mice. If you compare the spleens are quite small compared with the huge spleens in the liver. And if we wanted to know why it was working so good, our vaccine, we checked for the Th1 cytokine profile, and we observed that ours has a huge increase in IL-12 compared with the already published vaccine with the LLO peptide. And we also checked the specificity using tetramers, and we observed good percentages of both CD4 and CD8 interferon gamma producers. So protection with our dendritic loaded peptide with the GAPDH1-22 correlates with a good Th1 cytokine production and with a good ability to expand both types of specific cells, CD4 and CD8. So to conclude, why would it be important to have a listeria-based dendritic cell vaccine? Well, because there's no vaccine available for listeria, or in general for other intracellular bacteria, and because the listeriosis cases has increased tenfold in the last 5 years. And how is this? Well, usually you have in listeriosis 1-3 cases per year in every hospital, okay? And now we have 13, 14, even 15 cases every year in every hospital. So if you think, for instance, just here in Valencia, that we have 5 hospitals, well, there's a bunch of cases every single year. And why is this? Well, there's 2 reasons, or at least 2 reasons. We have made a change in our diet. We are eating much more prepared food, frozen food with long storage. And imagine that's just one bacteria of Listeria and a storage of the prepared food for 6 months. It will give you a lot of bacteria. And if you are pregnant, or if you are a little immunocompromised, or with your immune system a little bad, then you will have a problem with Listeria. And also because Listeria is an opportunistic pathogen in AIDS patients, and a new important thing in hospitals, transplantation patients. It turns out that this increase is because of the liver transplantation patients that has really increased in our health system, because we now have good health system for liver transplants, but the problem is that they get infected with Listeria. So that's the other reason. And there's a new field in Listeria, that is Listeria. It is right now prepared for cancer patients. In fact, there's one clinical phase in phase 1 and phase 2 using Listeria for all these different cancers. This is one company that is Advaxis, and this is the other company that is Aduro. And they have also a bunch of them. So this, in the future, they will probably benefit for a Listeria vaccine. And the people that make all this up, and thanks to all of you. Do you have any questions? Thank you.
Thank you very much. Now, this paper